Okay, good afternoon, everyone. Thanks to the organizers for having me at this conference. I'll be talking a little bit about the health beneficial properties of some berry species from Jamaica. So first, I'll talk about the research goal and objectives, then talk about the Jamaican context, show you some pictures of rubus berry species from Jamaica, share with you our methods and results, and finally end with a summary of our findings. So our research goal is to conduct research on uncommon Jamaican plant species aimed at investigating their health beneficial properties in order to promote productive utilization of the island's plant resources. And these are our research objectives. First, to determine the health beneficial properties of the fruit species, including antioxidant, anti-inflammatory, anti-cancer, anti-diabetic, and in the future, anti-hypertensive. We also carry out bioactivity guided approach studies, and we investigate the biological activity of the pure isolates and also try to disseminate our findings as widely as possible. So Jamaica is a small island comprised of 2.7 million people, about 7,400 square miles. We have 3,000 species of flowering plants. About a little under a third of that are endemic. However, quite a number of our plant species are unknown by the general populace. And one of the reasons is because they are grown in highly mountainous regions in the island, so they are not very easy to get to. And so because they are not generally known, they are not utilized as they could be. And the Statistical Institute of Jamaica in 2012 reported that over 600,000 pounds worth of fresh or frozen berries were imported into the island. All this happened when we have berry species that we have not at all utilized. So we're hoping that this kind of result will change that kind of dynamic. So there are 5 species of rubus fruits that grow in Jamaica. One is not shown here, and that one is endemic. These others are not endemic. That endemic one grows at the peak of Blue Mountain in Jamaica, the highest point, and that's why I haven't gotten to it as yet, but I will be looking for it. Okay. Yeah, no. So here we have a black raspberry species. These fruits are not fully ripe. Red raspberries, blackberry, and yellow raspberry species. Most of the work was done on these 3 species. In general, now, rubus hails from the Rosaceae plant family, and it has 12 subgenera and 250 species at least. And it's comprised of a wide variety of fruit types: raspberries, blackberries, dewberries, to name a few. And here we can see the rubus diversity going from yellow through red through black fruits. Now to our methodology and results. So we used several methods for antioxidant testing, one of which was lipid peroxidation inhibitory assay. And in this assay, we constructed a large unilamellar vesicle called a liposome. We did this using a glyceride, a phospholipid, and a fluorescent probe to make something looking like or functioning like a cell membrane. After we have our liposome, then we add iron 2, and that will cause oxidative breakdown of the liposome, and hence loss of fluorescence, which is measured spectrophotometrically. Any compound that will cause reduction in this loss is deemed to have antioxidant activity. So here are the results that we obtained on methanol, ethyl acetate, and hexane extracts of 3 berry species: the black raspberry, the red raspberry, and the blackberry species. So the ethyl acetate extracts were most active, had the highest antioxidant activity. And here we took those results from the ethyl acetate extracts and showed the fluorescent decay curve. This graph here is showing the DMSO control, showing what happens when iron is not added. So we're seeing that the liposome remains intact. This line here shows what happens when the iron is added without any antioxidant intervention. These two lines for R. racemosus and R. rosifolius, the red and black raspberries, show that they result in minimal destruction of the probe, and hence they have good antioxidant activity. For the blackberry species, there is initially noted destruction of the probe, but then you see that there's no further destruction over time. So these extracts are showing pretty good antioxidant activity. And we compared our extracts with extracts of the blackberry, the red raspberry, Rubus ideus, Rubus acuminatus, and a yellow cultivar of Rubus ideus. And these three are commercially available in the United States. And we're seeing that the species from Jamaica are looking pretty good compared to the ones commercially available internationally in terms of their hexane extracts. Where ethyl acetate extracts are concerned, though, the US species are looking better, though the blackberry species from Jamaica is quite comparable. And here we're seeing what the methanol extracts look like for those species. So we did cyclooxygenase enzyme inhibitory activity testing to test for anti-inflammatory activity of our extracts. Now, cyclooxygenase enzyme is important for normal cellular function. However, because it produces prostaglandins, which are associated with inflammation, any compound or substance that inhibits cyclooxygenase 1 and 2 would have demonstrated anti-inflammatory activity. So we use this as a test for anti-inflammatory activity. The positive controls we used were non-steroidal anti-inflammatory drugs, aspirin, Celebrex, and Vioxx, which is now off the market. The extracts from Jamaica showed low to moderate activity. Those from the United States did not show any activity in this test. The hexane extracts overall had greater activity than the ethyl acetate extracts. And the methanol extracts were not active. So we're talking about berries here, and it's hardly likely to talk about berries without talking about anthocyanins. So we're talking about the anthocyanins in the berries now. And these 5 anthocyanins were found in the berries via HPLC analysis. Cyanin 3-glucoside was the major anthocyanin found in most of the berries, except Rubus rosifolius, which has an orangish color, where pelargonidins were the major anthocyanins. So overall, R. acuminatus, the black raspberry from the United States, had the highest overall total anthocyanin content, but it was comparable to R. racemosus from Jamaica. All right, so we moved on to isolating compounds from the ethyl acetate extract, because that extract was shown to have the highest antioxidant activity. And we isolated 8 triterpenoid compounds of the 19-alpha hydroxyuracilic acid type via medium pressure liquid chromatography, preparative TLC, and trituration. And here are the compounds that were obtained, as well as the yields in which they were obtained. And these compounds here are the respective glycosides of the ones above. So 3 glycosides were obtained. And these are their LPO inhibitory activities, with compound 6 showing the greatest activity, followed by compound 2. The compounds also showed low to moderate anti-inflammatory activity, with compound 3 showing selective COX-1 inhibitory activity. We tested the compounds against some cancer cell lines indicated below, and compound 3 of note gave over 50% inhibition of the colon cancer cells. Compound 5 had the broadest spectrum of activity, however, the activity was rather low. And we did alpha-glucosidase inhibitory activity of compounds from the yellow raspberry as a means of anti-diabetic activity testing. And we used acarbose as a positive control, and this is a commercial anti-diabetic agent. And it's available in the US, Canada, and Europe and China under these names. So of note, compound 80D had half the IC50 value of this commercial agent, and so it's something of interest to do further research on. So we did quite a bit of research, and we're still doing research on rubus fruits from Jamaica, and we decided to move on to looking at the leaves as well, because it is well known that rubus leaves are pretty much non-toxic and they have good biological properties as well. So we looked at the leaves, we did fatty acid analysis, and we found that the omega-3 to omega-6 ratio was pretty good, 4:1, which is not usually seen very much in most plant sources. Also of note, the water extract of, and we're looking at the blackberries now, water extract showed very good antimicrobial activity with low minimum inhibitory concentrations against 4 bacterial species. The total phenolics and antioxidant activity of the leaf extracts was also determined, and we see that the methanol and water extracts were quite good, had good radical scavenging activity, and we used the DPPH assay here, and gallic acid equivalents for total phenolics, and the methanol extract was showed pretty high phenolic content. LCMSMS analysis revealed that epicatechin was one of the major compounds, phenolics present, followed by ellagic acid and catechin. However, this research is ongoing because we have isolated other phenolics from the leaves, which are they are major compounds, so we will be purifying them further and using them as standards to get a better picture of the phenolic profile in this plant. And we also did cytochrome P450 inhibitory activity, looking at the cytochrome, the 1A1 and 1B1 isozymes. And the reason why we chose these 2 isozymes is because they reduce the formation of procarcinogens. They're known to do that. So any compound that inhibits these enzymes will potentially have anti-cancer activities. The water extract, which before we noted had pretty good antimicrobial activity, is also looking good in this assay. So is the fat extract, the THG extract from the leaves. And so in summary, we have pioneered research on Jamaica's blackberry and raspberry fruit species and found that they have quite good health beneficial properties. The Jamaican berries compare very well with 3 species on the US market. Compounds from raspberry species demonstrate pretty good anti-diabetic properties, comparable or even better than acarbose. Compounds from Jamaican raspberries demonstrate a number of positive biological activities. The oil of Argemycensis leaves possesses 4:1 ratio of omega-3 to 6 fatty acids, and also the leaves had good, the water extract from the leaves as well as the fat extract had cytochrome P450 inhibitory activity. So I acknowledge the University of the West Indies in Jamaica for funding and the research team, Professor Nair from the Michigan State University, Dr. Malaba Galan Lu from the MSU as well, previously Dr. Navindra Siram, University of Rhode Island, and Tao Yuan, who works with him, and Yuan Zhang, Rupika Delgoda from UE, and these are collaborators that assisted with some of the assays. Thank you for your attention. These species grow wild. They're not cultivated. So at this point, they are present in small amounts at different locations of high elevation in Jamaica. We're hoping that within the next few years that will change significantly, where we will have cultivation, active cultivation and productive utilization via food processing, and so that we could have, rather than importing, we can be exporting products.
Have you done anything on dry berries, dried berries?
Well, most of the fruits that we've...
The nutritive value changes, right? So once we dry any berries, so have you done anything like that?
Most of the work we've done has been done on freeze-dried berries. So we freeze-dry them and then place them in the freezer and take from that. And we still are seeing good activity even after a few years in the freezer. But of course, upon processing for most food types, there will be some destruction or degradation of some biological component or chemical component. So, yeah, it would be less than the fresh, on a fresh weight basis for most things.
I've got lots of questions, but I'll just state one of them. How does the taste compare with
Okay.
species? I know some rubus species taste like absolutely nothing.
Yeah.
Others, like the raspberry, are very aromatic.
Right.
How would these species be on a scale from raspberry at one end and
Okay, so the black raspberry I find to be the most tasty, and I think it compares with the black raspberry that you get at supermarkets, which is this is the one we're seeing here. It's one of my favorites. I really like this one. Let me show you. This one is okay. It's not, I mean, the taste is not exceptional. And so it is for this. This one is quite good as well. This blackberry. It's just that it's small and the seeds are big relative to the size. So we actually we made a jam and juice from this fruit, and we showcased it at an agricultural show. People liked the flavor, however, they said the jam was too seedy, so we'd need to probably take out some of the seeds. So I think the taste isn't bad, but you know, as you said, berries, some of them are insipid, but others, I really love this one. Yeah. Welcome.
one more question? well, just one more, because for example, when you compare it with raspberry from the United States, you said, just how do you make sure that they have been treated in the same way between harvest and the time they arrive in your laboratory? Because things like the vitamin C content can change
Okay.
very much.
So some of them I actually went and picked, just like I picked the ones in Jamaica. And others, an associate of my supervisor at the time brought them in fresh. So.
Any other questions? And let's thank the speaker once more. Thank you very much.